Assalamu alaikum warahmatullahi wabarakatuh. I'm going to present my assignment in biopharmaceutical molecular absorption of proteins in gastrointestinal tract. At the first, what are proteins? Proteins are most abundant organic molecules of the living system. It provides the fundamental pieces of structure and function of the life. Proteins are macromolecules that consist of about 50% of the cellular weight. They are not only the cell building blocks, they also execute all the cell functions. Proteins are the polymers of amino acid. The functions of proteins. Proteins perform a variety of specialized and essential functions, static, structural, and dynamic functions, and the composition of protein are carbon, oxygen, nitrogen, hydrogen, and sulfur. The absorption of protein, it is inactive process that energy is derived from hydrolysis from ATP. This process occurs in small intestine. Absorption of amino acid is rapid in the duodenum and jejunum, but slow in the ileum. And the mechanism of amino acid absorption. There are two mechanisms of amino acid absorption. Number one, carrier protein transport system, and number two, glutathione transport system or glutamine cycle. The first, carrier protein transport system, the main system of amino acid absorption and active process that need energy derived from ATP molecule. There are 7 carrier protein, one of each group of amino acids. Each carrier protein has 2 sites for 4 amino acids and 1 of sodium. It co-transport amino acids and sodium from intestinal lumen to cytosol for intestinal mucosal cells. The absorption amino acid is to the portal circulation, while sodium is extruded out of the cells with exchange with potassium by sodium. The glutathione transport system announced a glutamine cycle where glutathione used to transport amino acid from intestinal lumen to cytosol intestinal mucosal cell. The energy needed is derived from ATP, and absorption of amino acid molecule need 3 ATP. Glutathione react with amino acid in the presence of glutacil, and glutamyl amino acid releases amino acid in the cytosol of intestinal mucosa cells with formation of 5-oxoproline that is used for regeneration of glutathione to begin another turn of the cycle. The absorption of intact protein and polypeptides. This period is a short period immediately after birth. The small intestine of infant can absorb intact proteins and polypeptides by endocytosis or pinocytosis. The infected proteins and polypeptides are not absorbed by the adult intestine. The macromolecule absorption in a certain individual appears to be responsible for antibody formation that often causes food allergy. The absorption of amino acid, the absorption occurs mainly in the small intestine. It is an energy required process. The di and tripeptidase, after being absorption, are hydrolyzed into free amino acid in the cytosol of epithelial cells. The activation of dipeptidase are high in the cells, L-amino acid are more rapidly absorbed than the amino acid. The transport of L-amino acid could occur by an active process, the amino acid by simple diffusion. Mechanism absorption of amino acid, sodium-dependent active process, and require ATP sodium diffusion along a concentration gradient. The amino acid also enters the intestinal cells. The sodium and amino acid share a common carriers and transport together. After that, compounds cytosine inhibit sodium-independent transport system. The challenges in the oral route of administration, the large molecule size, susceptibility to enzymatic degradation, short plasma half-life, high immunogenicity, the tendency to undergo denaturation, is bioavailability less than 1%. The other that have been tried with a variety degree of success are oral, buccal, intranasal, pulmonary, transdermal, and ocular. The potential problem in oral absorption, the oral administration of protein and peptide drugs faces two potential problems during their oral absorption. The first, metabolic degradation of therapeutic proteins and peptides by variety of proteolytic enzyme. Number two, poor membrane permeability. The pharmaceutical approach, chemistry modification of protein and peptide drugs improve their enzymatic stability and membrane permeation, and also minimize the immunogenicity. The prodrug approach include amino acid modification, the modification of individual amino acid by the substitution of the amino acid with amino acid can significantly alter physiological properties of proteins and peptides. For example, bismoglycin and the aminovasopressin. Hydrophobosation, the surface modification using the lipophilic meiosis, for example, Nobex insulin. The enzyme or protease inhibitors, the whole gastrointestinal tract and liver tend to metabolize proteins and peptides into smaller fragments of 2 to 10 amino acid within the help of variety of proteolytic enzyme. So protease inhibitors are co-administered with protein and peptides to alter the environmental for maximum enzyme stability to suppress proteolyte activity. For major type of enzyme, aspartic proteases, pepsin, or cysteine proteases, papain, or serine proteases, thrombin, or metalloproteases, carboxypeptides. The penetration enhancer are the formulation component of components that is important to dissolve the mucosal barrier to improve the permission of large macromolecule substituents like proteins and peptides. Following classes of compounds are commonly used: surfactants or polysorbate SLS, chelating agent, editor, fatty acids, sodium cabrates. Number 4, mucoadhesive polymers or thiomers, cellulose derivatives. Number 5, phospholipids or PC. Formulation vehicles, the oral delivery of therapeutic proteins and peptides can be successfully achieved by using dry emulsion, microsphere, liposome, and nanoparticle. The mucoadhesive polymers these systems prevent the pre-systemic metabolism of the therapeutic protein and peptides. For example, polymers, polyacrylic acid derivatives, and cellulose derivatives. The recent advances regulated or regulation, and number 2, evo form technology. Thank you.